immunotolerance, autoimmunity and hypersensitivity concepts. Hello friends, the topic of discussion here is basic concepts of immunotolerance, autoimmunity and hypersensitivity. To learn about these topics, it is important to get accustomed with these terms. So, let us look upon each of them. Starting with the history of immunotolerance. Normally, we do not make immune response against our own tissue, a concept known as self-tolerance. In 1945, R.D. Owen made a crucial observation that suggested that tolerance to self-antigens occurred because the immune system was exposed to these antigens during early development. He came to this conclusion after observing that adult twin cows each contained a mixture of their own and their twins' blood cells, indicating that they were equally tolerant of their own and each other's blood cell antigens. Exchange of blood between the two fetal cows occurred in the utero because binovular cattle share a placenta. This crucial observation demonstrated that tolerance could be induced by exposure to antigens in utero irrespective of whatever they were self or foreign. Shortly after this, F.M. Burnett enunciated the clonal selection theory, where he proposed that each lymphocyte was specific for only one antigen, and if a lymphocyte met this antigen during early development, it would be deleted from the repertoire. The theory was modified by J. Lederberg, who suggested correctly that it was the stage in the development or ontogeny of the lymphocyte that was critical rather than the stage in development of the animal. Moving ahead, the important aspect to discuss is about features of immunotolerance. Immunologic features of tolerance. Tolerance is different from non-specific immunosuppression and immunodeficiency. It is an active antigen dependent process in response to the antigen. Like immune response, tolerance is specific, and like immunological memory, it can exist in T cells, B cells or both, and like immunological memory, tolerance at the T cell level is longer lasting than tolerance at the B cell level. Induction of tolerance in T cells is easier and requires relatively smaller amounts of tolerogen than tolerance in B cells. Maintenance of immunological tolerance requires persistence of antigen. Tolerance can be broken naturally or artificially. Tolerance may be induced to all epitopes or only some epitopes on an antigen, and tolerance to a single antigen may exist at the B level or T cell level or at both levels. To further understand the concept of immunotolerance, let us now discuss about the mechanism of immunotolerance. Mechanisms of immunotolerance to self-antigens. The mechanism can be studied by understanding three phenomena. Let us now discuss about the mechanism of immunotolerance. Mechanism of immunotolerance to self-antigens. The mechanism can be studied by understanding three phenomena. First phenomena, that is central tolerance by clonal deletion. Central tolerance refers to mechanisms of tolerance acting during lymphocyte development in the thymus or bone marrow. Experimental studies shows that central tolerance is mostly due to the elimination or inactivation of those T and B cells that recognize self-antigens. These cells are destroyed or inactivated after they have expressed receptors for self-antigens and before they develop into fully immunocompetent lymphocytes. Deletion of self-reactive cells at any early stage in their development has been termed clonal abortion or clonal deletion. In early fetal life, self-reactive lymphocytes that are part of the developing immune system are deleted when exposed to self-antigens. As mentioned earlier, new immunologically component cells are generated throughout life, and these must be continuously deleted or inactivated also. Second phenomena is peripheral tolerance by deletion and inactivation. Peripheral tolerance refers to mechanisms acting on mature lymphocytes after they have left the primary lymphoid organs. Not all genes are expressed in the thymus, so developing T cells cannot be exposed to all self-antigens. Therefore, additional mechanisms for tolerizing self-reactive mature T cells are necessary. Mechanisms of peripheral B cell self-tolerance are also necessary because after simulation with antigen B cells expand and undergo somatic mutation, generating a population of B cells with new antigen specificities. Some of these cells may be specific for self-antigens. It is now known that at least 2 receptor ligand families play an important role in regulating T cell expansion in the periphery after contact with antigen, and these same molecules are probably involved in deleting self-reactive lymphocytes in the periphery. Peripheral deletion of CD4 plus T cells seems to be dependent on signaling through FAS. Mice and men deficient in this pathway fail to delete T cells and develop aggressive autoimmune disease. Sometimes T cells are not deleted but become specifically unresponsive to antigen stimulation, that is, they do not proliferate. This is called clonal energy. If antigen is removed, these energetic cells recover their responsiveness with time. One of the molecular mechanisms responsible for inducing energy is signaling via CTLA4, a molecule expressed by activated T cells. Mice deficient in this gene get a severe autoimmune disease, and there is some evidence that human autoimmune thyroid disease is linked to deficient expression. Now, the third phenomenon is suppressor T cells. The principal mechanisms of tolerance to self-antigens are clonal deletion and energy. It has been suggested that there may be a backup mechanism called suppression. This is a form of dominant tolerance because suppressor cells specific for a given antigen are able to inactivate other lymphocytes specific for the same antigen. For example, under some circumstances, it is possible to induce specific tolerance to allografts which appears to be maintained by suppressor T cells. Thus, when T cells are transferred from a tolerant animal, that is animal 1, into a synergic animal, that is animal 2, then they enable animal 2 to accept an identical allograft to the one given to animal 1. T cells specific for the allograft must still be present in animal 2 because if it does not receive T cells from animal 1, the graft is rejected rapidly. This suggests that the transferred cells from animal A are specifically suppressed the graft specific T cells in animal 2. The mechanism of action of suppression of this sort is not clear. Possible mechanisms could be the capacity of Th2 cells which secrete IL-4 and IL-10 to switch off pro-inflammatory Th1 cells. Moving ahead to the next concept, let's now have a look upon autoimmunity. How do we define autoimmunity? Autoimmunity can be defined as breakdown of mechanisms responsible for self-tolerance and induction of an immune response against components of the self. Such an immune response may not always be harmful. There are a number of causes that can be discussed which leads to autoimmunity. Causes can be categorized into release of sequestral antigen and immune stimulation. Let us first see the first cause of autoimmunity, that is release of sequestral antigen. Smoking can trigger Gout-Pasteur syndrome. Alveolar basement membrane normally not exposed to immune system. Smoking damages alveoli, exposes collagen. Anticollagen antigen damages lungs and kidney. Anti-sperm antibody produced in some men after vasectomy. Injection of myelin basic protein, that is MBP, produces MS-like EAE in mice, may be triggered by injury or infection. The second cause of autoimmunity, that is immune stimulation. Microbial infection stimulates APCs carrying self-antigen. High level of APCs with second signal breaks energy. Next thing that is important related to autoimmunity is its mechanism. Now, let us have a look at the mechanism. Antigen released from hidden location, antigen generated by molecular changes, molecular mimicry, alteration in antigen processing, infection, genetic factors, lymphocytes abnormalities, failure of central tolerance, overcome of peripheral tolerance and polyclonal lymphocyte activation. Effects of autoimmunity. First, that is tissue destruction. The example includes diabetes. Here, CTLs destroy insulin producing B cells in pancreas. The second is antibodies block normal function. The example include myasthenia gravis. Here, antibody binds acetylcholine receptors. Next, that is antibodies stimulate inappropriate function. The example is Graves' disease. Here, antibody binds TSH receptors. Another one mimics thyroid stimulating hormone, activates unregulated thyroid hormone production. Let us now have a look at autoimmune diseases. Insulin dependent diabetes mellitus, where pancreas is affected. Immune thrombocytopenic purpura, where the platelets are affected. Hemolytic anemia, where the target are red blood cells. Systemic lupus erythematosus, where number of organs like kidney, skin, joints, tendons, muscle, nerves, lungs, heart valves, GI tracts are affected. Graves' disease, where the thyroid is the target. Pernicious anemia, where the target is GI tract. Myasthenia gravis, where acetylcholine receptors on muscle cells are targeted. Ulcerative colitis, where GI tract is infected. Rheumatoid arthritis, where the attack organs are joints and other organs. Let us now have a look at different autoimmune diseases, where different types of autoantigens including cell receptors, cell membranes, proteins, hormones as well as total intracellular DNA and RNA are targeted. Systemic lupus erythematosus denoted by SLE, where whole intracellular DNA and RNA are targeted. Cell receptors autoantigens include three main diseases like Graves' disease, where thyrotropin receptors are present, myasthenia gravis, where acetylcholine receptors are targeted, and diabetes mellitus, where insulin receptors are targeted. The cell membrane autoantigens are in number of diseases like hemolytic anemia, where red blood cells are attacked, immune thrombocytopenic purpura, where platelets are attacked, and diabetes mellitus, where islet cells of Langerhans are attacked. Protein autoantigens are affected in rheumatoid arthritis and systemic lupus erythematosus, including immunoglobulins and complements. The hormones autoantigens are targeted in the diseases like diabetes mellitus, where insulin is affected, thyroiditis, where thyroid hormones are affected, and pernicious anemia, where intrinsic factors are affected. Now, how can this be cured? So, let us have a look at treatment for autoimmunity. Administration of immunosuppression, for example, prednisone and cyclosporin A. Removal of thymus, where some MG patients are removed. Plasmapheresis, which includes removal of antigen-antibody complexes. T-cell vaccination can be given to some patients where activated suppression of T cells is induced. By blocking MHC with similar peptide can also be practiced. Anti-CD4 monoclonal antibodies as well as anti-IL-2R monoclonal antibodies can be introduced. Having discussed about immunotolerance and autoimmunity, the next concept to be studied is hypersensitivity. Now, what is hypersensitivity reaction? Hypersensitivity reactions are harmful antigen specific immune responses which occur when an individual who has been primed by an innocuous antigen subsequently encounters the same antigen, produce tissue injury and dysfunction. Basic concepts of hypersensitivity includes allergen, the antigens that give rise to immediate hypersensitivity, atopy, the genetic predisposition to synthesize inappropriate levels of IgE specific for external allergens. There are basically 4 types of hypersensitivity reactions. They are 1, 2, 3 and 4. Gel and Combs classified hypersensitivity reactions into 4 types, studying each one separately starting with type 1. Type 1 hypersensitivity reaction. Of the 4 major hypersensitivity reaction, this has the most clear-cut immunopathological correlation. In the Gel-Combs conception, disorders such as hay fever or allergic asthma are classic examples of type 1 hypersensitivity. They implied that these disorders are driven predominantly by IgE bound to mast cells. On engagement of the cytophilic IgE with its appropriate antigen, mast cell degranulation and subsequent release of histamine causing an immediate reaction, leukotrienes resulting in the more delayed symptoms, and other mediators, the classic symptoms of allergic airway disease result. Type 2 hypersensitivity reaction. In the Gel-Combs formulation, type 2 hypersensitivity reaction are characterized by antigen-antibody interactions resulting in the local production of anaphylotoxin C5a, the recruitment of polymorphonuclear leukocytes, that is PMNs, and subsequent tissue injury due to the release of hydrolytic neutrophil enzymes after their autolysis. Classic examples of this kind of hypersensitivity reactions are the vasculitides. Type 3 hypersensitivity reaction. In the Gel-Combs system, type 3 hypersensitivity reactions occur when antibody reactions occur in the blood, resulting in the formation of antigen-antibody complexes which are deposited in the glomerular and pulmonary basement membranes. Here, the very presence of these complexes in addition to the PMNs attracted by the complement activation results in tissue injury and compromised function. Type 4 hypersensitivity reactions or cell mediated reactions. Gell and Combs conceived several organ specific autoimmune disorders as falling into type 4 hypersensitivity reactions. One example would be insulitis, seen in the early phases of insulin dependent diabetes mellitus. Here, the islets of Langerhans are infiltrated by a mixed cellular population comprising T cells, macrophages, natural killer, that is NK cells, and other leukocytes. Destruction of host cell ensures by a combination of apoptotic death and cytotoxicity. That's all about basic concepts of immunotolerance, autoimmunity and hypersensitivity. I end up my session here, and I am sure all that we have discussed here will be helpful to you. Thank you so much.
Summary of immunotolerance. Immunotolerance is an active antigen dependent process in response to the antigen. Tolerance may be induced to all epitopes or only some epitopes on an antigen, and tolerance to a single antigen may exist at the B cell level or T cell level or at both the levels. Mechanisms of immunotolerance to self antigens can be studied as central tolerance by clonal deletion, peripheral tolerance by deletion and inactivation, and suppressor T cells. Autoimmunity is breakdown of mechanisms responsible for self-tolerance and induction of an immune response against components of the self. Such an immune response may not always be harmful. Hypersensitivity reactions are harmful antigen-specific immune responses occurring when an individual who has been primed by an innocuous antigen subsequently encounters the same antigen, resulting in tissue injury and dysfunction. There are basically 4 types of hypersensitivity reactions. Types of hypersensitivity 1, 2, 3 and 4.